Hello, everyone, and welcome to Deep Dive. I'm Margaret Leinen, Director of Scripps Institution of Oceanography at UC San Diego. And in this series, I speak to scientists from Scripps Oceanography to take a deep look at their latest research. So with me today is Dr. Lihini Aluwihare, and she is a professor in the Geosciences Research Division at Scripps Oceanography. She is a chemical oceanographer, and she studies the cycling of carbon and nitrogen in the ocean. And her work focuses on how to read the messages that are encoded in molecules that maintain microbial life. And we'll definitely ask her about that. But also molecules that facilitate ecosystem interactions and contribute to the long-term carbon and nutrient storage in the ocean. Her tools also enable her to examine the human fingerprint on the chemistry of the ocean through detection of anthropogenic organic compounds. Lihini is also strongly committed to equity, diversity, and inclusion in geosciences in general and at UC San Diego. And she's worked across multiple levels of the university and in the broader national and international community to identify and combat institutionalized racism and sexism. So Lihini, can you tell us more about chemical oceanography and being a marine geochemist? What does chemical oceanography include?
So chemical oceanography is essentially, broadly speaking, it entails the study of the distribution of chemical elements in seawater. And we're trying to understand what are the sources of these chemicals, what are the sinks, the removal processes, and also what transforms them and how those transformations impact their lifetime in the ocean. And understanding kind of all these aspects of elemental cycling in the ocean allows us to understand or predict how these chemicals and their distributions in the ocean will respond to pulses and presses of change, things like global climate change. And so in this vast space, I've developed a particular interest in the part of chemical oceanography that's influenced by living organisms, and specifically how living microorganisms interact, as you said in your introduction, with the carbon and nitrogen cycles, and then how carbon and nitrogen moves back and forth between all the compartments, the water column, the particles, the organisms, and the sediments. I'm very fortunate because to do my research, I have to be in the water somewhere. And in coastal environments, we see both the complexity of how organisms modify their environment, but we also see the anthropogenic fingerprint, as you described. And then in open ocean environments, we can really observe how microorganisms structure their interactions in the absence of anthropogenic contaminants, but how they structure their interactions through the chemicals that they make.
So what kind of tools do you use to study carbon cycling?
Yeah, so we are, we use a lot of different tools. So one of the things that, we have to think about is just the complexity of these, as I said, I'm interested in carbon and nitrogen. So what's the complexity of the chemicals that participate in these cycles? And when we're thinking about the interaction of these cycles with organisms, you've got the vast complexity of what's genetically encoded. So we're talking about millions of different compounds, but once these organic molecules get into the ocean, they are transformed by light, they're transformed by their interaction with particles, with their interaction with different kinds of reactive chemical species. And so we know that this complexity is important. It enables organisms to perform their day-to-day activities. It enables them to interact with one another. And we know that these transformation processes are important in terms of determining how long these compounds might live in the environment. So sort of the storage capacity within this reservoir for carbon and nitrogen. So that's really the major challenge, right? How do we address this complexity? And a lot of my work has to do with developing advanced analytical methods to really kind of pick apart what are the molecules that are in there? What are their structural moieties? What makes them special? How might that allow us to understand what these molecules do in the environment, but also within organisms? Another aspect of that is the ability to use tools like stable and radioactive isotopes, because those give you a whole different view into how, what environments these compounds were produced in, how those compounds were transformed. And it's one of the things that I find really fascinating, that you can use isotopes of individual molecules in seawater to understand kind of the whole history of something. And within that space, there's also the use of radioactive isotopes. So using radioactive isotopes to get at the time scale. And that's really important when we're thinking about how the ocean responds to change. We need to understand rates at which these compartments interact with each other. And so radioactive isotopes really give us this additional power that we can't get with stable isotopes. So both of those together are things that I really enjoy applying.
So you used the tools in the conceptual sense. So most people think of tools as instruments and sensors and so forth. What kinds of infrastructure and instruments do you use to do the work?
Right. So, you know, I spoke of the complexity, which of course is the thing that I think about all the time. And some people think that my lab looks a little bit like a crime scene investigation TV show because we have some of the same instruments. Because it's, you know, you're still dealing with the complexity. You're trying to, you know, pick a particular signal out of all the different environmental signals the sample might have in a particular crime. And so we're thinking exactly about that. And one of the things that we might want to do, for example, is, you know, is tell the difference between something that might have come from a naturally produced, that might be naturally produced, and something that might be anthropogenic. So sometimes you can use the chemical handles to tell that. So for example, if we're thinking about something like DDT, we know that they have chlorine. It's a molecule that has chlorine in it. And so we might just look specifically for that handle. We might detect specifically for that. But then you might have compounds that can come from multiple sources. And that's when we can deploy tools like isotopes, because, you know, I spoke about radiocarbon. And if you have an anthropogenic compound in the environment, it's what we call radiocarbon dead. It's devoid of radiocarbon because it was synthesized essentially from petroleum, which is, you know, millions of years old, and the C14 has decayed away. Whereas something that's produced by an organism today in the environment has a different chemical, different isotope composition. One other way that we use radiocarbon was there's a gradient in the C14 content or the radiocarbon content of inorganic carbon with depth in the ocean. And so we use that gradient to actually understand how microorganisms in the deep ocean are making a living. So you isolate a particular molecule, because that's what we like to do, like DNA or a particular lipid, and then we can measure its isotope content and say, oh, this organism is getting its, even though it's living in the deep ocean, it's getting its food from the surface, or it's actually making its food in the environment. So both capitalizing on these advanced mass spectrometry tools that allow us to kind of unravel the complexity of these molecules, and then combining that with things like isotopes allows us to really get a very rich picture of not just what's there, but how those things are moving through the ocean system.
Can you give us an example of something that moves quickly versus and moves rapidly through the system versus something that sort of hangs around for a long time?
Yeah, that's a great question. It's actually what I spend my time thinking about. So you can think of you know, we often think of, when we think about fertilizer or something, we think about nitrate and phosphate, these inorganic components. But there's a lot of organic nutrients that are essential. So certain microorganisms, in fact, the most abundant microorganisms, have a very streamlined genome. So they don't actually have the capacity to make a lot of the metabolites that they need. So they might take organic sulfur compounds. Vitamin B12 is a great example. We don't think about that a lot, but yeah, a lot of phytoplankton don't make vitamin B12, so they rely on their microbial partners. And there's a lot of examples of molecules like that, and we're still learning, really, because we haven't been able to unravel the complexity. So vitamin B12 will be something that's very rapidly taken up from the environment. You're not going to find high concentrations of that sitting around in the water. Whereas some of these anthropogenic components, those are not, they are persistent by nature. And so we might expect them to accumulate on long time scales. And naturally speaking, you know, when we think about oil and, you know, especially oil and even coal, these are organic molecules that were transformed in particular ways, such that they're really, maybe they're not energetically favorable. You know, an organism, why would, you know, in a fairly desert-like ocean, why would they expend the energy to eat something that's not going to give them much in return? And so we see compounds like this, and that's actually a huge part of what I'm trying to really, this is sort of my discovery part that I've been working on for my whole career, is really trying to describe what it is that, what kind of chemical signature would tell us whether an organic compound is good to eat. You know, people use like the analogy of steak and a tire or something like that. You know, what are the chemical signatures that make something a tire, you know, that nobody wants to eat? So those are some examples.
Or ice cream in a tire.
Yeah, that's a better one.
So you said that many people said your lab looks like a crime scene investigation. So that brings us back to this idea of human fingerprints. So in your crime scene investigation, what have you found as you looked for those fingerprints?
Yeah, it's been really interesting because, you know, when you go into this coastal environment, you have all these different signatures that you see. You know, we collect the samples, we analyze them with our tools, and there's all this information. And so how do we start to unravel some of these questions? So actually, I've been thinking about this very recently because I was born in Sri Lanka, and yesterday or 2 days ago, a container ship caught fire outside the port of Colombo, which is the major city there. And I know a very famous marine biologist there, Asha Devas, and she called me and she said, what do you think is all the impacts of this? And so she started telling me what was in the container. So there were all these, you know, there was acids, but then there was cosmetics. And then of course there's the fuel oil, right? And so, you know, one of the things we have to think about, you know, is what are, you know, what do we as humans put into this environment, and what are the impacts on these ecosystems? How do those things even get into the environment in the first place? And so, you know, a lot of the things that we found are what you would expect to find, things like persistent organic pollutants. So the DDT suite of compounds is an example, PCBs, these brominated compounds, these flame retardants that were manufactured until very recently. But then you also start to find things like cosmetics or drugs, or, you know, during Christmas time, you find cinnamon and vanilla, right, in the wastewater. So, you know, our connection to the environment is so profound, and you see that even expressed in these at the individual molecule level. But one thing that we also discovered, or that, you know, others have also discussed this, is that, you know, a lot of the times when we're looking for anthropogenic signatures, we're monitoring particular things. So taking the DDT example, my collaborator, Unha Ho at SDSU, she was leading this charge. And what we found when we looked at different samples of dolphin blubber from the local region was not only that it contained, you know, over 300 compounds that were of anthropogenic origin, but it's only a fraction of those compounds are actually monitored or actually being detected. And the same with the DDT suite of compounds. There were more than 45 compounds that we could connect back to DDT, either through the manufacturing process And so if you were to take sort of a, if you put the blinders on and you're only looking for certain things, that's all you're going to find, right? And so one of our really innovations has been trying to do what we call this untargeted approach, really trying to get a snapshot of the whole, which is not easy. And we had a lot of pushback because it's hard to come up with even big data analysis methods to really sift through all this information. But it really gives you a profoundly different different insight into what's going on. And just to go back to the DDT question, just because it's an interesting example, there are certain chemicals that are that we think are included in the technical mixture that are in pretty small amounts, but when we go and look in the blubber of these organisms like marine mammals, where they've you've gone through many steps in the trophic food web, we see them in relatively higher concentrations. And so that suggests to us that they're being biomagnified for some reason, or maybe they're being transformed, something is being transformed and turning it into what's in the technical mixture. But if we didn't look for that, or if we didn't find that, we wouldn't know that it's having a kind of it's not a parallel pathway, right? It's sort of doing something a little bit different than some of the other DDT compounds. And if we really want to understand what impact these chemicals can have in the environment, we really need to be able to look at them on a molecule by molecule basis, because their structures are different and so they behave differently through the system. So that's another reason it's really important to be able to look at the whole instead of just kind of it's like, the analogy everybody uses is the blind man and the elephants, right? If you only touch the tail, what would you know? So yeah.
So you mentioned that you look in marine mammal blubber. Why do you look in the blubber?
Yeah, because it smells really good. No, it was actually, when we were doing this project, it was very sad. This fin whale washed up in actually on Mission Beach. And so because we were connected to this project, we were allowed to come and take a sample. And that's the first time I've actually ever been in a place at a site like that, where you've got, they basically, it was huge, right? So they had to bring bulldozers to pull the animal on shore, and then they were cutting up the blubber, and that was, I'm glad that I only got 1 gram blubber samples for the other analyses, because it wasn't that fun we literally had like slabs. We had like a 50 kilogram slab of blubber from the fin whale. So the blubber is, one reason we look at marine mammals is, of course, they are at the top of the food chain. So they're apex predators, and we specifically want animals that also eat other fish. So, if you're feeding pretty low on the food web, you're not going to accumulate these compounds to a great extent. So one reason we look at them is because they're apex predators. And then the blubber is a place where a lot of these anthropogenic contaminants accumulate, because they're, this is a question people often ask about DDT related compounds. They're sticky. They don't want to be dissolved in water. So they're going to stick to particles. And in an organism, they're going to partition into the most fatty tissues that they can find. And so the blubber is the place where these things really accumulate. And if we want to get sort of a more comprehensive picture of the anthropogenic fingerprint in an environment, that's a great place to go, because a lot of these compounds are present in high enough concentrations that we can actually detect them.
So you mentioned DDT and then briefly mentioned flame retardants. Are there other so I think people are familiar with the fact that both of those have been banned. Are there other organics, human generated organics that you see regularly that are used?
Yeah, so I think so there has been a ban on the perfluorinated compounds, the PFAs, and we see a lot of interest in those, because they are also incredibly persistent, and they are in the waste, they get discharged in the wastewater. So we see that in the coastal environment. There's another.
What do they come from?
They are used. Oh, that's it. Oh, why can't I? Sorry, I have a brain. I'm trying to think. They're used in industrial manufacturing, but I think they're sort of they use them as a coating for something, but I have to look it up. I can't remember, sorry. And I was just talking about this because we just have it in a paper, but just and so there's so there are PFAs, and we see it a lot, but one of the concerns is in drinking water, but we see it in the wastewater. And then there are some other compounds that we've been seeing lately too, that different versions of these, they're aromatic compounds through their benzene related compounds that are used in the manufacturing of the rubber in tires. And it's a sort of an emerging contaminant. It's been detected in a variety of different places, and some of the chemicals that are found in these tires actually are transformed through photochemistry in the water to form dioxins, which are very potent. They're known to be some of the most potent dioxins are known to be some of the most potent compounds in terms of their toxicity. And those have been shown, they've been linked to they've been linked to growth inhibition in salmon. And so those are actually becoming emerging contaminants as well. And so those are two of the ones that seem to have some currently a lot of interest in them, because they're being detected at relatively regular frequency in a lot of these water monitoring endeavors. And we see all of them in our our time series off the pier, for example.
So you've been talking about DDT a lot, and just recently we found that there are many barrels of what we think are barrels that we think are full of DDT offshore of California, and some papers published about that. And how is this also something that you're working on, and how has that affected your research?
Yeah, it's something that we're definitely thinking about and involved in. When we first did the characterization of the DDT suite of compounds in the dolphins, we know of the historical Superfund site off of Palos Verdes, where Montrose, basically discharged through the White Points outfall DDT into the sediments. And so we were familiar with that dump site. And, the bottom line is that DDT is in our current food web, right? So regardless of what the Superfund site has done, there's DDT in our food web. And so we originally, we assumed that was the main source. And we also know, people use the DDT concentration in marine mammals that migrate all over the world as a indicator of the fact that they feed off of Southern California, because we're talking about orders of magnitude higher than any other location, at least in the US, for DDT. So this rediscovery of the potential for at least comparable amounts of DDT being in the sediments off in this deep dumping site, the dump site 2, I think, in San Pedro Basin, that makes us wonder, okay, well, maybe there's another input of DDT into the food web. And what's been interesting to us is that, in our dolphin study, we looked at inshore feeding dolphins and offshore feeding dolphins, and you don't see a significant trend in terms of concentration or body burden between those populations, whereas you do see it for some of the other things like PCBs, for example. And so it's not to say for sure that that's a smoking gun, but that was something that we had recognized previously, and several of Unha's papers subsequently has also noted that. And so that might, that raises the possibility that in fact the DDT in the offshore site is entering the food web. And I think that's always something we've wondered, because the DDT does, it will end up in the sediments. So how it gets into the food web is really an important question to answer. But the other thing that we wonder is whether there's, the DDT barrels, or at least what's purportedly dumped there, is the waste from the industrial manufacturing process. And so, there could be a pretty unique fingerprint associated with how that material is moving through the food web. And that's something we're really interested in, because we have these innovative tools that can kind of, really where the complexity is accessible, we can capitalize on that to really try to get sort of a fingerprint of the suite of compounds that might be present in those sediments in the deep site versus in Palos Verdes, to try to see how those, whether those fingerprints show up differently in the food web in different locations. So that's what we hope to do, is to be able to characterize the fingerprint of what's in the sediments, or hopefully even if we could get access to what's in the barrels, and see if that fingerprint tells us something different than what we would expect to see from the Palos Verdes site. Yeah.
As you think about chemical oceanography and your area of chemical oceanography, and look into the future, sort of 10 years, beyond 10 years, whatever, what are the big opportunities that you see there? What do you think that people will be doing with this in the future?
Yeah, that's a great question. And there are lots of different types of chemical oceanographers who probably prioritize very different things from me. But, one of the reasons I got into this field was because of the importance of organic matter in the carbon cycle, right? Whether it's transport of carbon in sinking particles out of the euphotic zone, whether it's these, we talked about these long-lived versus short-lived compounds. And so I think, we talk a lot about the factors that influence how much its carbon is fixed by phytoplankton in the ocean. But I'm really interested in what impacts kind of the removal of it. What are the environmental and community composition level changes that impact the respiration rate of carbon, whether it's in the surface ocean, but also in these low oxygen environments. And there's a lot of renewed interest in these low oxygen environments, especially in the water column, because there's, with climate change, there's the possibility, and there's some evidence that suggests that they would expand. And so how microorganisms function in those environments to respire carbon versus, allow it basically, by not working on it, to preserve in the environment, is something that I'm interested in studying, and very and we have some work going on in those areas. I think another big question is the fate of terrestrial carbon as it comes into the ocean. And that's also not a new problem, but now that we see how land use change and warming in the high latitudes has mobilized a lot of this carbon, bringing it into coastal environments, this is a very important question. We really need to quantify how much of that carbon is being turned into CO2 or methane and released into the atmosphere, essentially, acting as a positive feedback to the warming, versus how much of it is either getting exported into the deep ocean or being transformed in some way. And so I think that again is a problem that lends itself well to some of the tools that I'm developing, because again, we have to be able to look at molecular level changes. And then we also want to couple with isotopes, because some of these carbon stores are actually quite old. And so we should be able to differentiate those from using radiocarbon techniques. So I think those are two, looking at the way that respiration rates in the ocean are being impacted by climate change and changes in oxygen concentration, and then the fate of terrestrial carbon. And I think this is an important thing to also consider, because we know that things like peat, but also wetland carbon, is a very potentially really important future source of carbon sequestration, or locations where we can sequester carbon, because they actually have a really high capacity, these waterlogged sediments, both in peat, tropical peat, and these coastal sediments. So trying to understand what determines the respiration rate of that carbon and how to tweak that to increase storage, I think has ramifications that go beyond just the discussion of terrestrial carbon transport.
So at the beginning of our conversation, I mentioned that you have done really so much work to help us diversify Scripps. Why is diversity important for science?
Right, I mean,
it's important socially, right?
Right.
Is it important for science?
Yeah, I think, the easy answer, and I think it is a correct answer, which is that having a more diverse group of people involved in the enterprise means that the type of products will be different, right? The kind of science that we do. But I think it goes beyond that, right? If we don't have people with the lived experiences, the cultural sensitivities, and the cultural competency that underrepresented individuals bring to science, there are going to be problems that we aren't even thinking about as being important, right? As we, the climate problem is a great example, right? Any climate solution that doesn't center the dignity of poor people or the dignity of Black and Brown people is not a climate solution. And if we don't, many of us can read books and become educated, but the lived experiences and the cultural competency is crucial, not just to identify what kinds of scientific questions and how to do science differently, right? I think that's a key thing that we have to keep in mind. Science has been done in a particular way because of the people that were involved in it. And as we change who gets to get a seat at the table, science is going to evolve, and it will evolve in ways that are probably very relevant for what's happening in the world today. But it also means that we can train the next generation of scientists differently. And so I think it's not just about, having diverse minds work on a problem might come up with diverse solutions, but it's a transformation of what we will be doing as scientists going forward. And it's really, I think it's difficult work. There's no H index, for that kind of effort. And it's uncomfortable. It makes people uncomfortable, but it's really important that we think about what it's going to take for us to evolve into the 21st century as earth scientists and ocean scientists.
For any non-academics out there, what's the H index?
Well, it's how many people read your papers, essentially. That's basically what it is. So, yeah.
Yeah, it seems to me that it also, having diversity in science and in the voices that we listen to is also important for how well people trust the science. Mm-hmm. If it's not relevant to you, or if science has overlooked major issues that you see as being relevant, then you may say, well, they don't even know this. Why should I trust them?
No, that's exactly right. And I think that's, you articulated that well. And that's what, that's why I think the climate solution issue is such, this is why it's so important to involve all these different voices, because that will require everyone to come along, right? That will, and the rolling out the COVID vaccines, that's another great example of where it's really important. I mean, and in health sciences, we've really seen that play out, right? In a lot of different ways, where it hasn't been very inclusive. That has influenced what kinds of, medical conditions we focus on, but it's also influenced how efficiently we can convince people to trust the system, as you put it, with these vaccines. So yeah, it's a really good example of why inclusivity is important.
So I'm enjoying looking at the photographs behind you. And I see children, and I see some beautiful art. And but I don't see any pictures of dogs, and I'm surprised.
Well, yeah,
Everyone at Scripps knows that you
I guess
foster.
there's one.
Oh, there's the dog. So everyone at Scripps knows that you are the foster mother supreme for dogs. How did you get into that?
Yeah, so there was a former graduate student at Scripps, Ashley Corrigan, who runs the rescue that I work for. It's called the Rescued Dog. And you're lucky I'm not at home today because I have 5 puppies and they're very vocal. I'm fostering them right now. And I lost my longtime dog who's actually in that picture. And I had said to my husband that, I was going to go to the pound and find a dog, and, I'm going to bring it home. And even if it attacks my only child, Rookshawn, I was still going to keep it because that's what we do. And so he thought he was, we were having this conversation. And so one of my former graduate students, Janan, suggested that I look into fostering, and she put me in touch with Ashley. And so that's how I first got into fostering dogs about 7 years ago. And and I've been, one of the things I found really funny was that the first dog I fostered, I decided to adopt him. And so they call you this foster failure. And I was immediately offended by that. And I was like, no, I don't fail. That's not going to happen. And so it was kind of like, it was for my pride that I continued to foster. And I'm glad I did, because I really enjoyed it. And it just gives me, it really changes my frame of mind when I go home and sit in a pile of puppies, even if they're covered in poop.
Well, that's organic too.
Yeah, exactly.
That's part of the grand cycle.
Right.
All those microbes.
I'm glad you understand. Not many people do. Yeah.
Well, as you and I have said before, the microbes rule the world. They just let us live in it because we're great hosts for them.
Yes. Yes, indeed.
Oh, Lihini, thank you so much. It's always a pleasure to talk to you. And thank you for taking the time to do this little film so other people can enjoy a conversation with you and understand the really interesting things that you can do with chemistry and with organic chemistry in the ocean, and that it's really important for us. You're looking at these things that are very toxic and can move into the environment, and as you said, can be in our food chain. And we certainly don't want to be thinking about eating DDT. It's bad enough to have it around us.
Yeah.
And also, thanks for all of your work on enhancing diversity, equity, and inclusion at Scripps, at UC San Diego, and in that field that we both love so much, geosciences.
Thank you. Thanks for this opportunity. It was really wonderful. Thank you.
Great. Thanks everybody for joining us for another deep dive with Professor Lihini Aluwihare.